another way. Isolation chip or I chip, that is called as one of the methods that would help cultivation, in situ cultivation of the uncultured bacteria. So it is high throughput method. So what you are going to do is you are going to dip a plate with multiple holes in the plate that you are going to have, and you are going to put it into a suspension of the sample, which is environmental sample. You are going to have a mix of those different types of bacteria and organisms into it. So all of them are going to get captured onto the plate, but they are going to get captured as one cell in one hole, because the hole size, the hole dimensions are such that it will accommodate only a single cell. So it is like the filtration assembly, or you can say the straining assembly, and a single particle only will get retained in a one pore. So when you talk about eye chip assembly, you are going to have membranes which are going to cover the holes from either side. The hole is that of a disc, right? And disc or plate, you can say, depends on the shape you are going to have. And there are pores or the holes in on the plate or the disc, and on both the sides you are going to apply the membranes. So upper and bottom plates, you are going to have with matching hole press, and the membranes are going to be loaded onto the plate, right? And with the help of screws, you are going to tighten the assembly. There are going to be the compartments, three parts. We will see it in the next diagram how it really looks like. So you are going to have the tray, you are going to have the plate on which you are bore the holes. The tray also is going to have similar size pores, and then you are going to have the filter. So these are the three components, and it is something like diffusion chamber again, but the extended form and more number of pores are there. So here in the diffusion chamber, all the bacteria, those which are going to be inoculated onto the surface of the gel, would be growing, and the number is limited considering the size of the diffusion chamber. But here, because the plate is having so many pores, and each pore is going to act like as a culture vessel, so many bacterial types or cells can be entrapped in at a time simultaneously. At the same time, they are well isolated and separate. So this is the design of the eye chip. Here, this is a horizontal diagram. If you see part C, there are three layers, right? And if you observe it clearly, the each of these layers is having a horizontal plate, and on the plate you have the pores, uh, porous plate it is, right? And on the top of that you are putting the filter, right? So in this case there are 384 holes in one chamber, and each is about 1 millimeter in diameter. So the chamber is 1 millimeter diameter, huh? And in that you have having hundreds of pores, so it is micro size pores you are going to have. So each is going to be like a standard petri plate or single chamber, like, in which the bacteria will get entrapped and grow. So as the size of the individual cultivation chamber decreases, the diffusion with the surrounding environment would increase, right? And it will give you a kind of a better culture condition, and maybe you are going to get a better throughput, better efficiency, high number of bacteria, types of bacteria maybe growing in it, because interaction with the natural medium, the nutrient medium, is much better than that. So microfluidics based cultivation, miniature, that is what it is called as. So you are going to handle the cells as individual cells. They are getting incubated in the micropores, and each one is a pure, so picoliter of the volume of the nutrient media will be passing through it, and you can just harvest one pore as one plate, equivalent to one pore. So when you harvest, it is a microcolony developed from a single cell, and then you can transfer it to maybe 98 well plate, which will be again used as a culture method, right? Fine, I think we shall stop with this.